The focus of this video is mitochondria, the powerhouse of the cell. Mitochondria are subcellular organelles. They're probably one of the most familiar of all the organelles, so that if someone shows you a picture of an animal cell, you can probably look at it and pick out the mitochondria. Look, here's one. Here's another one. Same thing is true for plant cells. We can look at a plant cell and say, yep, there's a mitochondria in there. Oh, here are two more. The fact that so many people are familiar with mitochondria reflects the fact that they're really important. They're most famous for their role in energy production. Mitochondria are the site of the TCA cycle and of oxidative phosphorylation, which is going to allow the cell to maximize ATP production. You should know, though, that mitochondria also play roles as calcium stores, so they maintain calcium levels in the cytoplasm. They can play a role in programmed cell death. They're involved in the production of steroid hormones. They can deactivate compounds in nerve cells. They can deactivate some neurotransmitters. In liver cells, they can be involved in processing ammonia. They can also generate heat to maintain body temperature in mammals and birds, and so they really serve a number of very important functions. Now, although cartoons like this are really good for giving us an overall sense for what's going on in the cell, they're not very accurate representations. Let me show you what I mean with respect to mitochondria by showing you some images from real cells. The first image I'm showing you here, this is from a HeLa cell. That's a human cell line. It's derived from a cervical cancer, and these cells have been stained with three different dyes. The blue is a dye called DAPI, which stains the nucleus. The green is a cytoskeletal marker, and then red, that's stained with a dye called Mitochondria. And what I want you to notice is that there are lots and lots of small ovoid structures. Each one of those is a mitochondrion, and there might be hundreds present in this image. Here's an image of another cell type called PC12 cells. These were originally derived from a rat tumor. When these cells are grown in culture under the right conditions, they take on a neuron-like appearance. They send out these processes that look like dendrites or axons that extend from the cell body, and we can see that in this image here. Well, once again, the cell's been stained with three dyes: DAPI for the nucleus, green to stain tubulin, and then red for the mitochondria. And what I want you to notice again is that there are a lot of cells in the cell body, but look, here we also see mitochondria in regions that would correspond to axon terminals, or perhaps the dendrites. And we can also see that the mitochondria are closely associated with the cytoskeleton, and in fact the cell will transport mitochondria along microtubules to get them to a region where they need energy. Well, I hope the two images that I've shown you have done a good job of conveying that there are many mitochondria in cells. I can't give you an absolute number for how many mitochondria are found in a cell, because this varies from cell type to cell type and organism to organism. Different types of cells contain different numbers of mitochondria, with more metabolically active cells such as muscle and nerve cells and liver cells containing more mitochondria than less active cells. For example, heart muscle cells have a whopping 40% of their cell volume devoted to mitochondria, while in the lung epithelial cells, mitochondria make up less than 1% of the cell volume. The number of mitochondria also differs between species, so if we compare the same tissue, say liver, in a mammal and a reptile, the mammal will have a higher number of mitochondria and a greater intermitochondrial membrane surface area than the lizard does. And these differences are even evident within mammals, with smaller animals like mice having more mitochondria than larger animals like horses. Mitochondria are very small organelles, so while we can see that they're present in bright field and fluorescence microscopy, we can't see their structure. To learn more about the structure, you have to use electron microscopy. Well, here's an electron microscopy image taken from lung tissue showing us two mitochondria. And what I want you to notice is that each one of these mitochondria has two lines around the outside. It has a double membrane structure. This double membrane structure is thought to reflect the endosymbiotic origin of mitochondria. You may already be familiar with this theory. What it posits is that a long time ago, as cells were evolving, some cells took on what we would characterize as a eukaryotic shape, with nuclei and other organelles like endoplasmic reticulum. But these primitive eukaryotes were anaerobic. They lacked the ability to get energy using the electron transport chain. At the same time, however, there were prokaryotic organisms, bacteria, that had the ability to do photosynthesis and aerobic respiration. What happened then, about 1.5 million years ago, is that one of these anaerobic eukaryotes ingested an aerobic bacterium And rather than consume it completely and degrade it, that bacterium was able to take up residence and eventually then turned into a mitochondrion. So with respect to the double membrane, the inner mitochondrial membrane then would reflect the aerobic bacteria membrane, whereas the outer mitochondrial membrane would resemble the plasma membrane, which engulfed the bacterium when it was taken into the cell. Chloroplasts are also thought to derive through an endosymbiotic origin, so that in a second event, sometime later, one of these aerobic eukaryotes then ingested a photosynthetic bacterium, and that then evolved to become a modern photosynthetic eukaryote with chloroplasts. And chloroplasts also have an inner and an outer chloroplast membrane, reflecting this origin. There are some other pieces of evidence supporting the endosymbiotic origin of mitochondria. For example, we know that mitochondria and chloroplasts have their own DNA and that it's circular, like bacterial DNA is. We also know that mitochondria will divide by fission. They pinch and divide into two parts, and that's the way that many bacteria reproduce as well. Leaving that aside for now, let's return to the electron microscopy images. Well, here we are, and for emphasis, I've colored in the outer and the inner mitochondrial membranes with yellow. It's a little hard to see in electron micrographs, so I'm going to turn it into a cartoon, and now we can look at the structure in more detail. Just to orient you, here is the cytoplasm, here's the outer mitochondrial membrane, the inner mitochondrial membrane. There's a space between called the intermembrane space, and then within the inner mitochondrial membrane, that space is called the matrix. This cartoon illustrates that mitochondria are compartmentalized, and there are these 4 different compartments. I want to talk about them a little bit one by one. Let's begin with the outer mitochondrial membrane. Well, this is an unfolded membrane, and it is similar in composition to other membranes in the cell. You might have noticed that I drew it as a dashed line, and that's because it's relatively permeable to the cytoplasm. The reason that it's permeable is because there are large complexes called porins, which allow ions and small molecules to move freely between the cytoplasm and the intermembrane space. Larger molecules like ATP or proteins are too big to pass through porin, and so there are also many transporters for these larger molecules. The outer mitochondrial membrane is also rich in enzymes needed for mitochondrial membrane synthesis, because if a mitochondrion is going to divide, it's going to need more membrane. Let's look at the inner mitochondrial membrane. Well, this membrane has characteristic infoldings called cristae. They're finger-like projections that extend into the matrix. The lipid composition of the inner mitochondrial membrane is different from others in the cell. Some lipids found in the inner mitochondrial membrane are uncommon in other membranes in the cell, but they are found in prokaryotic membranes. An example of this would be the lipid cardiolipin. Cardiolipin can make up to 20% of the lipids in the inner mitochondrial membrane of mitochondria, but we don't see it elsewhere in the cell. It got its name cardiolipin because it was first discovered in heart cells. And I'll just remind you that heart cells have a very high concentration of mitochondria, but really this is a little bit of a misnomer, because really, with the exception of the inner mitochondrial membrane, it's way more common in bacteria than it is in mammalian cells. The inner mitochondrial membrane contains many proteins. Something like 60 or 70% of the weight of the membrane is made up of membrane proteins. We can broadly divide these into 2 categories. There are transporter molecules and enzymes that are necessary for a variety of metabolic functions. Unlike the outer mitochondrial membrane, the inner mitochondrial membrane is not permeable, and so we're going to need transporter molecules in order to move substances across that barrier and into the matrix, or out of the matrix to the rest of the cell. The second broad class of proteins found in the inner mitochondrial membrane are these large complexes which are involved in the electron transport chain and then the FO-F1 ATP synthase. Together, the electron transport chain and the ATP synthase are going to allow mitochondria to maximize ATP production in the cell. The third mitochondrial compartment is the intermembrane space. It's similar in ion composition to the cytoplasm, although it does contain a distinct set of proteins, some of which are only found in the intermembrane space. One example would be cytochrome C. And last but not least, we've got the matrix. The matrix contains hundreds of enzymes necessary for the tricarboxylic acid cycle, lipid metabolism, and other metabolic pathways. It also contains the mitochondrial DNA and mitochondrial ribosomes. It's a very crowded and viscous environment, and you can see the difference in density when you look at electron micrographs. And finally, Unlike the rest of the cell, this is a part of the cell that is basic. It has a high pH with a very low concentration of protons. That pH gradient is generated by the activity of the electron transport chain and is key to ATP synthesis. I'm going to close this video by showing you some electron micrographs. Remember that earlier in the video I said that cells had the ability to place mitochondria at different locations in the cell so that they could meet local ATP demands. Here's an example from a skeletal muscle cell. The inset in the upper right corner is a lower magnification and then a higher magnification view on the full screen. And what we're looking at here is a longitudinal section through skeletal muscle fibers. Remember that muscles contain actin and myosin, and that's involved in muscle contraction. The actin and the myosin are organized into these long structures made up of repeating sarcomeres. That's what the S stands for. And that's the part of the cell that will actually contract. So here we're looking at one long contractile element. Here's another long contractile element. There's another one that I didn't color above it. Lying in between these bands of sarcomeres, we find mitochondria. I'll color some in with red. Muscular contraction mediated by actin and myosin requires ATP, and here the cell has placed mitochondria right next to these contractile elements where it will be needed. Here's another example. This is a picture of sperm. In the top left, we see a fluorescently stained image. Once again, the DNA has been stained blue. Here, the cytoskeletal elements necessary to whip the flagellar tail are red, and then green is mitotracker. The image underneath shows us what this would look like with a transmission electron micrograph, where we'd be looking at the head containing the genetic material and then the start of that flagellum. And here's a blow-up of that region showing you that wrapped around the flagellum there's a high concentration of mitochondria. Again, for sperm to be able to navigate and swim and do flagellar motion, they'll need ATP. In this last image, the sperm has entered the egg and the head is beginning to dissolve and the genetic material is being released into the cell. And again, we can see on either side of that shaft some mitochondria. Well, I hope this overview of mitochondria will serve useful as you continue in cell biology and look in more detail at many of the processes in which mitochondria function.